Hello everyone, welcome back to my YouTube channel. Today I am going to explain the structure and composition of lysosomes. Okay, so lysosomes are an animal cell or digestive organelles. Lysosome are membrane enclosed organelles that contain array of enzymes. Typically, lysosome contains at least 50 to 60 different hydrolytic enzymes which are capable of breaking down all types of biological polymers such as proteins, nucleic acids, carbohydrates and lipids. So these are the all enzymes and they catalyze or active on the substrate, okay, particular substrate, act on the particular substrate. The all digestive enzyme are produced in the ER, okay, so and transport the lysosome, which were discussed in my previous video. So please check the description box for more details. Okay, so lysosome function is the digestive system of the cell, serving both the degraded material take up from outside of the cells and to digest obsolete components, that means outdated components of the cell itself. Okay, lysosomes variable size. So you can see in this picture, you can see that Kupffer cells of the liver showing at least 10 different lysosomes of in highly variable size. For example, this is the one, this is the largest, okay, then two smallest, then smallest, okay, 3, 4, 5, total 10 considerable variation in size and shape of lysosome. So why it is regular shape and size of lysosome? Because they taken up different material, okay, that have been taken up for the digestion. That's how difficult it is to identify these organelles on the basis of morphology. Lysosomal having acid hydrolysis enzymes. This is the structure of lysosome. Okay, you can see here this is the acid hydrolysis enzyme within the lumen of lysosome, and this is plasma membrane. Okay, this is pH should be 4.6 to 5. So they are all enzymes, lysosomal all enzymes share an important properties. Okay, for example, all lysosomal enzymes are acid hydrolysis, which are active at the acidic pH, that is the 4.6 to 5, and that must be maintained within lysosome. Okay, acid hydrolysis enzyme would be inactive at the neutral pH of the cytosol, that is the pH 7. Now that's why to maintain that acidic internal pH, lysosome must actively concentrate the hydrogen ions. Okay. So how it happen? Okay, how it accomplished? Okay, that accomplished by a proton pump in the lysosomal membrane, which actively transfer proton into the lysosome from the cytosol. So this pumping requires expenditure energy in the form of ATP hydrolysis. Okay, you see here the ATP. Okay, they hydrolyze and ATP they convert into the ADP. Then lysosomal membrane. Okay, so this is one of the most important characteristic of lysosomal membrane. They contain the variety of highly glycosylated proteins. Integral proteins, okay, highly glycosylated integral proteins. Glycosylated proteins, that means they proteins they adding the carbohydrate chain, which were discussed in my previous video. Please check the description box for more details and watch the video. Okay, so carbohydrate chain are thought to form a protective lining, okay, that shield the membrane from attack by the enclosed enzyme, that is the acid hydrolysis enzyme. Okay, so next it is the what is the lysosomal storage disease. So if the mutation in the genes that encode enzymes, okay, are responsible for more than 30 different human genetic disease, which are called the lysosomal storage disease. Okay, so why it is lysosomal storage disease? So the undegradable material accumulate within the lysosome of affected individuals. So there are main two types of lysosomal disease. Here I describe. One is the Gaucher disease. In Gaucher disease is the most common disease. Why it is? Because the mutation in the gene that encode lysosomal enzymes, okay, required for the breakdown of glycolipids. One. Second one is the eye cell disease. So deficiency in the enzyme that catalyzes the first step in the tagging of lysosomal enzyme with the mannose-6-phosphate in the Golgi apparatus. As earlier described in my previous video, that lysosomal protein first must be glycosylated, okay, that carbohydrate chain, okay, in carbohydrate chain that mannose molecules are present which which is phosphorylated, okay, mannose 6 phosphate in the Golgi apparatus. Okay, so this is all about today's video. uh, I hope it is helpful to you. Thank you for watching.